Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 for January 2021. This is Part 2. I already did the Part 1 video which had from question 1 to question 4 of this paper. I will put the link to that video in the description box. Let's begin with this part. And again, Part 2 contains question 5 to question 8. Let us begin. Question 5 says, photosynthesis consists of the light-dependent reactions and the light-independent reactions. In the light-dependent reactions, light energy is converted into energy stored in ATP. Explain why light energy is converted into energy stored in ATP. Now, the key thing we need to remember here is light comes from the sun, or the energy comes in the form of light. However, the plant itself cannot directly use the light, so the light has to be converted into a different version of energy, which is ATP, which the plant can then use to drive cellular processes. So I said light energy cannot be directly used by the plant. Conversion of a light energy to ATP provides a form of energy usable. This is usable. ATP is usable by the plant for the light-independent reactions of the Calvin cycle in order to convert GP into GALP, or you can call this GALP. So this is extra information I added, but the key thing is the plant cannot use sunlight or light the way it is to drive cell processes or like metabolism and so on. We will need ATP because this is the version or the usable energy that the plant can use. So the next part says, an equation can summarize the production and breakdown of ATP. Complete this equation by writing the names of the substrates and the type of reactions on the three dotted line provided, the three dotted lines. So we have this line, and that line. If we can see here, this is ATP, and they have told you this process is phosphorylation, the process leading to the other side. If that is phosphorylation, it means formation of ATP. And ATP is formed if we combine ADP and the inorganic phosphate, you can call it PI. So ADP plus PI produces an ATP molecule, and here a water molecule is lost. So it means to reverse that process, we need to add water. That is why this process is called hydrolysis. Hydrolysis, water is added to an ATP molecule, and then it's broken down into ADP and PI, which is the inorganic phosphate. Let's continue. Explain the role of light energy in the light-dependent reactions. Light energy is used to excite electrons from the chlorophyll molecules, and these electrons are going to be used for phosphorylation as they travel from higher energy level through either through the cyclic photophosphorylation or the non-cyclic photophosphorylation. They will be used in the formation of ATP. And then the light is also going to be used to carry out photolysis of water. Here, the electrons are going to be provided that can replace the electrons that were lost from the chlorophyll molecules. Also, the hydrogen ions that are going to be used in the reduction of NADP. So the ATP and the reduced NADP are therefore going to be used in the light-independent reactions of the Calvin cycle. So let's continue. The next part says, the light-independent reactions use products of the light-dependent reactions to produce simple sugars. Which row of the table shows the products of the light-dependent reactions that are used in the light-independent reactions? So let's see, here they say for ATP production, ATP can be produced by both non-cyclic and cyclic photophosphorylation, but in this case we can see you could go with that. Since we are looking at the product which is going to be NADP, it has to be in the reduced version, and in this case, it has to be non-cyclic photophosphorylation, because in this case, the non-cyclic photophosphorylation will produce ATP and the reduced NADP, which can later go into the Calvin cycle. So it should be D as the answer for this question. So let's continue. Here they say simple sugars have the formula CnH2n or N. Name the organic molecule from which each element in a simple sugar originated. So carbon, of course, is going to come from carbon dioxide. And again, remember, for formation of these sugars, we need carbon dioxide and we need water. The carbon dioxide will provide the oxygen as well as the carbon, and the water will provide the hydrogen. So that is how these come to be, or that is how the plant is able to get them in order to use them for the formation of the simple sugars. Roman 3 says simple sugars are used in the synthesis of new biological molecules, which row in the table shows the inorganic ions that are needed to synthesize the new biological molecules. If we are making a protein, of course, we'll need carbon, oxygen, nitrogen. We'll need nitrates. Nitrogen is going to come from nitrates, and some proteins, of course, will have some sulfur, but since it's not here, I will just go with nitrates. Now, RNA should have phosphates as well as nitrates because RNA has those nucleotides, and remember, they have a nitrogen base and they have the phosphates, so they should have phosphates as well as nitrates. And then triglyceride, this one should have neither nitrates. It can never be made out of nitrates nor out of the phosphates. So because a triglyceride is purely made up of carbon, hydrogen, and oxygen, there is no nitrogen, there is no phosphorus, and there is no sulfur. It's only that. So we can just say neither that nor that. This is the end of question 5. Let us continue to question 6. Question 6. The photograph shows 2 zebras. We can see this and that. Those are 2 zebras. They say the stripes of zebras are thought to reduce attack by biting flies. Biting flies cause stress and disease to cattle, reducing food production and causing financial losses for farmers. So let's continue. They say, A, farmers use chemicals to kill the flies. Flies often evolve resistance to a new chemical within about 10 years. Explain how flies evolve resistance to new chemicals. If there is a resistance that occurs to the new chemical, it's because the DNA has been modified, and that could be due to a random mutation. So when a mutation occurs, the DNA of these flies has maybe received a new allele, I would say, so formation of a new allele, and this allele is going to lead to resistance against the new chemical within these flies. So it means the flies that have the new allele will not be affected by the chemical, while those that do not have the allele will be affected by the chemical and therefore die. So in this case, the chemical is acting as selection pressure, killing those that do not have the allele, while those that have the allele are going to be able to survive. So the flies with the allele that allow resistance to chemical will survive, and they will reproduce, and when they do, they will pass on their allele to their offspring, and their offspring will also have that ability to be resistant to the specific chemical that they're using to kill the other flies. And over time or over generations, every fly that will be available will be resistant to that specific chemical. And that is how the resistance to the chemical, or the evolution, evolving resistance to the new chemical is going to be able to occur. So I'll continue to the next part. This part says, suggest why the evolution of resistance to these chemicals occurs so quickly in flies. Now, what we need to know, it occurs quickly because flies have a very short life cycle. Their life cycle is really short, so they produce a lot of offsprings, and it means within a shorter period of time, they're going to have so many offsprings, they're going to have so many generations down the line, and if there is any mutation, it's going to develop rapidly, and the effects from this mutation are going to be seen within a shorter period of time. So I say, because the life cycle of flies is short, these flies produce a lot of offsprings. This is very important, and some are bound to develop mutations leading to new alleles. Having many offsprings, there is a lot of changes that can occur. Imagine if one fly had maybe 100 offsprings. One of them is going to develop a mutation, maybe one of them is going to develop a mutation when exposed to this chemical, and when that happens, there is a chance of producing offsprings that could have the specific mutation, giving them a selective advantage and therefore ability to survive under the harsh conditions. The next part says, an investigation was carried out to see if the presence of stripes on cattle reduced the number of biting flies on their bodies. 3 groups of black cattle were compared: unpainted cattle, cattle painted with black and white stripes, and cattle painted with black stripes only. The cattle repel flies by flicking their tails, stamping their feet, and skin twitching. So the frequencies of these fly repelling behaviors were recorded. The graph and table show some of the results of this investigation. So we're going to look at the graph first. On the vertical side, we have the mean number of biting flies on body and legs of cattle. So they were counting them. And remember, this is the mean number, telling us that the experiment was repeated and the mean was calculated. You can see we have error bars here. It shows that they basically calculated the mean, and the mean was compared with the lowest and the highest value. So let's see, here we have the unpainted cars, unpainted cows or cattle. You can see they had more mean number of flies. Those that were painted black and white, you can see they had a little bit lower in comparison to the others. And the black stripes also had a little bit higher, but in a little bit lower than those that were unpainted. So these are the cattle, and then here we can see the mean number. So I will continue to the next page. Here we have another table. The table shows the group of cattle. Here, the unpainted, black and white stripes, and those with black stripes. And here we have the number of flies on the body. Here we have the number of flies on the legs. So we can see this one has the highest number of flies on the body. And here we can see the highest number of flies on the legs. There is another table here, table 2. We see the groups of cattle still as they showed us here. But in this case, they are looking at the flicking, the mean frequency of fly repelling behavior, number per hour, the flicking of their tails. So here we see the unpainted had a 54, while those that are black and white had 55, and so on. So we can look at the data in the table. Of course, we're going to be calculating the percentages, any percentage reduction, percentage increase, and so on, in order to be able to answer the next question. So here they say, determine the effectiveness of painting stripes on cattle in reducing the number of biting flies on them, and use the information in the graph and both tables to support your answer. Remember, they are talking about effectiveness, so we have to know which method is more effective, or are they all effective or not. So I took them step by step. I began with a graph. So based on the graph, again, I want to take you back to the graph so that you can see it carefully. And this time, the values I calculated now can make sense. You can see this comparison here. We can see from here to here, we see a 13%. And this time to this one, we can see a 55%. That is going to be the change that has occurred. So I'm going to reference to these values in my answer on the next page. So let's go back. They say, I said, based on the graph, that's the graph we have just seen, painting the body of a cattle with either black or white, black or black and white stripes reduces the number of biting flies on the body and the legs of the cattle. You saw the decreases, and I calculated those percentages. So we saw here, so on the cattle, or legs as well as the body as well as the legs of the cattle. And then I said black and white stripes lead to greater reduction in the number of biting flies. Here we saw there were only 58 flies, and that was a 55% reduction. While those that were black stripes, we saw 111 flies, meaning there was a 13% reduction. Also, you remember we saw there were error bars, and the values had the mean values had been calculated. So I said, however, because the error bars of the unpainted overlap with those of black and white stripes, the results may not be significant enough to be used in determining effectiveness of painting stripes on cattle. Can I take you back again to the previous graph so that you can see what I'm talking about? These are the error bars we're talking about. You can see this error bar here and that do overlap. So these results here may not be that consistent or may not be that valid if you're trying to use them to measure effectiveness. This will agree with that, and this will agree with that because the error bars are not overlapping. However, that and that tells you that maybe this method is not that good. It's for being used to measure effectiveness or determine effectiveness of the method you have chosen. So I will go to the next part. So here I talked about the two tables, table 1 as well as table 2. So table 1, I said from table 2, painting the cattle with black and white stripes has no effect since the results were similar to those of the unpainted cattle. Again, to take you back here, we can see painting black and white. You can see that and that. It's as if they're kind of the same, not that different. So it may not be that effective to paint them black and white. So I also went on to say painting with black and white stripes produced a decrease in stamping of feet from 16 to 10. That is about a 38% reduction. However, the skin twitching increased from 5 to 8, so there was an increase by 60%. Also, painting black and white stripes has very little effect on flicking of the cow's tail, and there was a slight decrease. from 54 to 42, which is about 22%. And again, calculating percentages can sometimes help you earn extra marks. Usually, if data has some mathematical information, you can use it to calculate and show the examiner that you're good with when you work with numbers. So I went on to the last table, which is table 1. I say table 1 shows that black stripes reduce the number of biting flies on the legs by 21%. However, these were not effective in reducing the number of flies on the body. And again, let me just do this. They were not effective. So let's continue. I also say there was a 92.3% increase in the number of flies on the body. This is just extra information. And lastly, when you paint with black and white stripes, it reduced the number of flies on the body by 65% and the number of flies on the legs by 46%. This shows that painting with black and white stripes is more effective than painting with only black stripes. So in this case, we can see there is effectiveness based on the results from table 1, and also based on some of the results, we can see it creates better results. However, the reliability of the data will not be valid if, like we saw from the graph, if there is overlapping of the data set, then it will not be that reliable. So this brings us to the end of question 6. So let's continue on to question 7. Question 7, gel electrophoresis is used to separate DNA fragments of different lengths. The rate at which the DNA fragments move through the gel depends on several factors, including the molecular size of the DNA fragment, the shape of the DNA fragment, and the concentration of the gel. So all these are going to determine how the fragment moves through the gel. So part A1 says, which enzyme is used to cut DNF into fragments? The enzyme is called a restriction enzyme. This is used for DNA synthesis, that is used to insert a DNA segment into other DNA, and then this is reverse transcriptase for converting RNA into a DNA. So for synthesis of DNA using RNA. Part Roman 2 says, explain why the use of an enzyme to cut the DNA results in fragments of different lengths that can be separated by gel electrophoresis. So when we use DNA, or when DNA is being fragmented, we are using restriction enzymes. Restriction enzymes have specific sequences or base sequences on the DNA molecule that they do recognize, and when they do recognize those recognition sites, they recognize, and they are not evenly placed along the DNA molecule, so they're going to be at different sites or different lengths along the DNA molecule, and when they are cut, we're going to have fragments that are not the same length. Also, the enzyme will carry out hydrolysis of the phosphodiester bond at an equally placed recognition sites, resulting in the different sizes of the fragments, and that's why they move at different distances along the gel. And again, remember, a restriction enzyme is used along the DNA molecule. Those recognition sites are going to be placed differently. Maybe if one is, let's assume if one is, 2 centimeters from the other, another one could be 10 centimeters away, so the fragments are not going to be the same length. And again, the numbers I've referred to are just hypothetical numbers. The 2 centimeters and 5, those are hypothetical values, so please don't take them the way I said it. I was just using them as an example. So let's go to the next part. Part B says, fragments of double-stranded DNA move through the gel at a relative rate. They've called the rate MR. That is inversely proportional to the log of their molecular weight, MR. So they say, complete the table using the equation that. So remember here they've given us MW. We want to find the MR. So we'll just feed the MW here and use your calculator. 1 divided by the log of, this is 100,000. When you use your calculator, it will give us 0.2. And also, when we do the reverse, since we are given 0.34, that is this value here, and then we want to find the MR, we'll find 873 as the answer. Just input into your calculator, and then it will be able to give you whatever you're looking for. So the next part says, the diagram shows the position of a DNA fragment with an MW of 10,000 after gel electrophoresis. Complete the diagram to show the position of a DNA fragment with an MW of 100,000. Use the information in the question. Now here, it's very imperative that you know that this is the origin is here. This part here is the origin. If you know that this is the origin, then you should know that the DNA is moving in this direction. Let me use, it's moving in this direction. So because it's moving in that direction, and you should know that heavier fragments are going to move slower. So if this one has a 10,000, the one with 100,000 is going to be far away behind, closer to the starting point or closer to the origin in comparison to the one that has 10,000. So I placed it around this point, not too close and not too, kind of a little bit far away from this place. So they will allow any position within this range here, provided it's not meeting up with this one. They're not at the same level, and provided it's not touching this. So provided it's within this region, it will be acceptable for you to just mark that. So that was not hard to do. It was just really easy. Let me go to the next part. Here they say the fragments move more slowly through a higher concentration of gel. Suggest why the fragments move more slowly through a higher concentration of gel. Now, the higher concentration of the gel is going to give resistance to the moving parts, or the gel is going to have a higher resistance. So it means these fragments that are actually heavy are going to be moving through, and their speed is going to be affected because when the gel is thick, it means the molecules within the gel are tightly packed together, and there are smaller spaces for these fragments of the DNA to move through. So it's going to be a little bit harder for them to move through the thick gel, meaning the speed at which they move is going to be very low or very slow. So they'll be moving slowly because of the tightly packed molecules within the gel. The next part says circular DNA moves at a faster rate through the gel than linear DNA. Give 2 examples of circular DNA found in cells. We have plasmid DNA, we have the mitochondrial DNA, we have the chloroplast DNA, and we have the bacterial genome, or you call the chromosome or nucleoid of the bacteria. So this part says give 2 differences between the structure of circular DNA and that of linear DNA other than their shape. Now, linear DNA has unbound phosphate. As you know, the deoxyribose, it's unbound phosphate as well as deoxyribose, while circular DNA does not. The linear DNA has the 3' and the 5' ends, while the circular DNA does not have those. And then the linear DNA is more associated with the histones. It binds tightly on the histones so that it can be folded, and while the circular DNA does not. There is also one fewer phosphodiester bond in linear DNA than in circular DNA, which with the same number of mononucleotides. So if they have the same number of mononucleotides, there is going to be one fewer phosphodiester bond in linear DNA than in the circular DNA. So this brings us to the end of question 7. Let's continue to question 8. Question 8, queens are fruits that grow on trees. When a queen falls from the tree, 2 processes take place: browning and decomposition. Browning takes place when the fruit is cut and exposed to the air. Browning is catalyzed by the enzyme polyphenol oxidase, or you can call it PPO. The photograph shows browning in a queen's cut in half. So let's see here. Here we can see the fresh cut queens showing some browning, and then cut queens exposed to the air showing extensive browning, that one here. So let's continue with the question. They say activity of the enzyme PPO at different temperatures was investigated, and the graph shows the results of this investigation. So we have the substrate concentration on the y-axis, and we have the time after start of the reaction on the x-axis. So we can see the substrate concentration, of course, is decreasing over time at different temperatures. However, the rate at which the substrate concentration decreases is different depending on the temperature that is used. We can see here at a temperature of 40 degrees, we can see there is rapid decrease, and then it slowly kind of levels off. Here at 50, we can see there is no rapid decrease. It means maybe there is denaturation that is beginning to be formed. So the rate at which the substrate concentration is decreasing is not as the one we have at 40 degrees. So let's go to the next page to see some of the questions. And again, do ignore this part here. I'll come back to this. I'm going to use those in my calculations a little bit later. So the first part says, sketch the graph for enzyme activity plotted against temperature for this enzyme. They say use the data from the investigation. Remember what we have here. It was about 40. The optimum optimum temperature was about 40 something there. So we expect the enzyme activity to increase and increase and increase until the optimum, and then its activity is going to decrease, as you can see. So anything you draw like that will be correct. And the next part says, calculate the Q10 for this enzyme using the data from this investigation shown in the first graph. So they say use this formula. Q10 is equal to RT plus 10 divided by RT, where RT is the initial rate of the reaction at T, and the RT plus 10 is the initial rate at T plus 10. So here I wrote it there, and I said using RT as the initial rate at 30 degrees. So I took the one at 30 degrees. Remember RT, they've told you that at T is going to be, if T is, the RT is going to be T plus 10 degrees. So if I use my RT to be 30, it means I'm going to use RT plus 10 to be 40 degrees. So that's what I chose. So at 30 degrees, the rate of RT, like they say here, initial rate, RT. Again, I'll take you back just a minute. From my graph, I chose 30 and 40. You can see at 30 degrees, there is this line here. I found the gradient of that line to be the rate, initial rate. Since this line is going to be the tangent at the initial part, basically. It's a straight line, so I just find the gradient of that line. And then for 40 degrees, I'm going to find the gradient of this line because it's going to tell me the initial rate for that reaction. So using this and that to find the gradient of this line, and using that point and that point to find the gradient of that other line, I can be able to find RT and RT plus 10. So my RT is at 30 degrees, which is 43 minus 36 divided by 2 minus 4, giving me that. And my RT plus 10 is 40 minus 34 divided by 1.9 minus 3, which gives me this. Now, when I go back here, my Q10 should be negative 5.5 divided by negative 3.5, giving me a 1.57, and this is my answer here. So let us continue to the next part. B. A quince fruit is made up of cells and contains a lot of juice. The table shows the composition of carbohydrates in the juice of one species of quince. So here we have the carbohydrates. They've given us the monosaccharides, the disaccharides, trisaccharides, and tetrasaccharides. So we have the different monosaccharides. However, the disaccharide is only sucrose, trisaccharide is only raffinose, and then here we have the tetrasaccharide. Ignore this part in red. I'm going to use it for my next calculation. I'll use it to show you some question we're going to answer as part of the next questions that follow. So the first part says, complete the table to show the ratio of concentration of the 4 types of carbohydrates. So 4 types of carbohydrate, it means they want us to find the ratio of monosaccharides to disaccharides to trisaccharides to tetrasaccharides. So all I need to do is, since I already have the total of the tetra, the total of the tri, and the total of the di, I need to add up all the monos to get the total of the monosaccharides. So adding up all the monosaccharides, it gives me 1,361.3 milligrams per 100 centimeters cubed of juice, and that is the total for all the monosaccharides. And here the ratio is going to be that to that to that to that. When I divide through by the small x, which is this, I get a 164.012, a 6.868, a 1, and a 1.145. Now here I need to find a whole number ratio. So this is going to be 164. When I round this off, it's going to be a 7, and that is going to be a 1, and this is going to be a 1. I was looking for whole number ratios, so this is what we call, this is my answer here, after making sure that these are all whole numbers. So let me go to the next page. So here they ask which row of the table shows the carbohydrates with the median concentration and the model concentration in quince fruit. So I want to go back slightly behind. Remember, we are looking for the median and we are looking for the mode. It's very easy to see the mode because the mode will have the highest number that is repeating. So 8.3 and 8.3, this is going to be the mode number. And to find the median, I have to arrange them in increasing order. So since this is the smallest, then you come to that and that. Or you could even say this and that are the same, so you can call this 1 and 1, and then you skip to go to 3. Actually, let me do like that. Let me just call this 1 as well, 1, 1, and then I'll skip to 3. So 1, 1, skip to 3, and then come back to 4. There is a 5, there is a 6, 7, 8, 9. So when you cancel out, cancel 1 and cancel the 9, cancel the 2, the other 1, and cancel the 8, cancel the 3, and cancel the 7, cancel the 4, and cancel the 6, you remain with a 5. That is going to be the median, the number that is left when you arrange everything in increasing order and then you cancel out on both sides. So 5, so the median is going to be basically this 57.0, while the mod is going to be 8.3. So when I come here, and again to take you back, 8.3 gives us, 8.3 is for either inositol or it's going to be for raffinose, and then the other part is going to be for sucrose. So we have to look at the table that gives us both of them. So since they only gave us raffinose and sucrose, so I just go with that, which is D. The next question says, explain the role of decomposition in returning the carbon present in carbohydrates in the quince fruit back into the atmosphere. Use the information given in part B to support your answer. So decomposition, of course, the decomposers, which include the fungi and the bacteria, will release enzymes onto the substrate or the carbohydrates that are to be broken down, which are present in the quince fruit. These enzymes will digest through hydrolysis processes, producing monosaccharides as they break down the glycosidic bonds. So for tetrasaccharides, here we remember tetrasaccharides are going to have 3 glycosidic bonds, so these are going to be broken using 3 water molecules. And for trisaccharides, 2 glycosidic bonds are present, so we'll break them using 2 water molecules. And of course, for disaccharides, they have 1 glycosidic bond, so it's going to be broken using 1 water molecule. So for example, remember they taught us to use examples from the table. Sucrose is going to be broken down into the monosaccharides, which are glucose and fructose, and these monosaccharides can be taken up by the bacteria into their cells by diffusion as raw materials or as sugars that are going to be used for respiration. Let's continue. When cellular respiration occurs, it could be aerobic or anaerobic. When it occurs, byproducts like carbon dioxide are going to be produced, and these are going to be released to the atmosphere as gases or as CO2 gas back to the atmosphere. So that's how the whole process occurs. So this brings us to the end of this part of this paper. Thank you for being with us, and I hope to see you in our next video. Please do not forget to subscribe to our channel. And as I said previously, I will put the link to the first video or the video part 1 of this paper in the description box so that you guys can check it out. See you in the next paper. Bye-bye.